First pathway lecture after the flipped carbohydrate structure class. Students arrive, sugar chemistry, aldose versus ketose, epimers, the glycosidic bond. Today you'll use that vocabulary. Glucose goes to pyruvate in 10 steps, and the 3 irreversible ones are where all the regulation and all the disease live. So we use 3 stages. There's glucose to F16 bisphosphate cleavage, then G3P to pyruvate. The Leninger figure prints only 2 because it groups 1 first, 2 together. So mark on your notes where the 2 ATP are spent, where the 4 are made, and where the molecule splits. Red accents mark the irreversible steps. We spend ATP to make ATP. One kinetic footnote you will meet again. Hexokinase and glucokinase catalyze the identical chemical reaction, but they are regulated in opposite directions and live in different tissues. Hexokinase shuts itself off when the cell is full. Glucokinase does not. The liver keeps taking up glucose after a meal for as long as the portal vein keeps delivering it. Its activity is instead restrained by a separate glucokinase regulatory protein that sequesters the enzyme in the nucleus when glucose is low and releases it when glucose rises. 3 more points on this slide, each of them examinable. 1. This is a regulated, irreversible step. Control point number 1. 2. Hexokinase is inhibited by its own product, glucose 6-phosphate. Glucokinase is not. Keep that difference in mind. 3. Glucose 6-phosphate is a branch point. It can go on through glycolysis. It can go into glycogen, or it can go into the pentose phosphate pathway. Glycolysis is one of several options, not an inevitability. The clearest single payoff of the flipped class was learning about the isomers. Here it is. Step 2. Glucose 6-phosphate becomes fructose 6-phosphate. It's catalyzed by phosphoglucose isomerase. It's reversible. The delta gene is approximately 0, so no ATP is involved, no regulation, no disease. What it does chemically, an aldose becomes a ketose. The carbonyl moves from carbon 1 to carbon 2. Glucose and fructose are constitutional isomers. Same molecular formula, different connectivity, and this enzyme performs that interconvertible conversion in one step. Why it matters? Because moving the carbonyl to carbon 2 creates a symmetry that the aldolase needs 2 reactions later to cut the molecule cleanly in half. This step is a setup for the split. Here is the most important reaction in the lecture. This is the reaction. Step 3. And this is the most important reaction in the lecture. Fructose 6-phosphate plus ATP becomes fructose 1,6-bisphosphate. The enzyme is phosphofructokinase 1. It is irreversible. It costs your second ATP and is the rate-limiting step of glycolysis. It is also the committed step. Everything upstream of phosphofructokinase 1 had other destinations available. You could go to glycogen pentose phosphate pathway. Past this enzyme, the carbon is going to pyruvate. That is what committed means. And that is why the cell puts its main control here rather than at step 1. The regulators. You have to memorize these. They are asked directly. Phosphofructokinase 1 is inhibited by ATP and by citrate. It is activated by AMP and by fructose 2,6-bisphosphate, which is the most potent activator of the two. Insulin and glucagon exert their control over this pathway through fructose 2,6-bisphosphate, acting at this enzyme. 2 more facts. Dietary fructose enters the pathway below this step, thus escaping this control. And deficiency of the muscle isoform of phosphofructokinase causes Teruri disease, the glycogen storage disease type 7. Contrast that, we'll talk about the glycogen storage diseases later. So, why 2 ATPs? Keep that in mind. Aldolase cleaves the 6-carbon fructose 1,6-bisphosphate into 2 3-carbon phosphate dihydroxyacetone phosphate, which is a ketose, and glyceraldehyde 3-phosphate, which is an aldose. This is where the 2 ATPs you spend pay off. Each 3-carbon half now carries one of the phosphates you added. That is why both phosphorylations were necessary, one for each half. Neither triose could continue without one. Okay, so this is the point. This is where the pathway doubles. From here to the end, every reaction runs twice per glucose. Every yield number downstream is a per triose number that you have to multiply by 2. The exam point on this slide is the isoforms. Aldolase A is in the muscle and red blood cells. Aldolase B is in the liver and kidney. Aldolase C is in brain. All 3 cleave fructose 1,6-bisphosphate, but aldolase B has a second job. It also cleaves fructose 1-phosphate, which is the intermediate in dietary fructose metabolism. It is that second activity that is lost in hereditary fructose intolerance. Same enzyme, different substrate, and the disease comes from the substrate you would not think to associate with glycolysis. Okay, so stop and take stock. 2 ATP gone, 2 G3P. Everything from step 6 is profit, and it runs twice. Triose phosphate isomerase converts dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, so that both halves of the original glucose can enter stage 3. This is reversible. Without this enzyme, half the carbon of every glucose molecule would be stranded as dihydroxyacetone phosphate. The yield would be cut in half. It is often called a catalytically perfect enzyme. It works as fast as diffusion allows. One clinical footnote: the reaction intermediate can break down to methyl glyoxal, which is toxic, and the enzyme speed is partly about committing the molecule before that side reaction can occur. 2 ATP have been spent. You're holding 2 molecules of glyceraldehyde 3-phosphate. You are 2 ATP in the whole. Everything from step 6 onward is recovery and then profit. And all of it runs twice. The arsenate story earns 30 seconds. It proves the acyl phosphate is what carries the energy forward. Glyceraldehyde 3-phosphate becomes 1,3-bisphosphoglycerate. The enzyme is glyceraldehyde 3-phosphate dehydrogenase. It's reversible, runs twice. 2 things happen in one reaction. An oxidation, NAD plus is reduced to NADH, and a phosphorylation, inorganic phosphate, not ATP, is attached, creating a high-energy acyl phosphate bond. This is the only oxidation in glycolysis. Both molecules of NADH per glucose come from this single step, 1 per triose. If an exam question asks where the NADH comes from, it is here, and there is no second source. So this gets the constraints the rest of the pathway. NAD plus is present in the cell in limited amounts. If it is not regenerated, this step stops. And if this step stops, glycolysis stops. Everything we say later about lactate and about the shuttles is a solution to that one problem. Notice what this step consumes: inorganic phosphate. Under conditions of rapid glycolysis, the availability of free phosphate can itself become limiting, which is exactly the mechanism you'll see when we discuss hereditary fructose intolerance, where phosphate is trapped and the cell effectively starves the pathway of a substrate it never thinks about. 2 attachments. First, arsenate. Arsenate substitutes for inorganic phosphate at this step, and the product then hydrolyzes spontaneously. The oxidation still happens, but the high energy bond is lost as heat, so no ATP results. Arsenate uncouples this step. That is the classic phrasing. Second, in the red blood cell, the product of this reaction is a substrate for the 2,3-bisphosphoglycerate shunt, which we'll cover later. Okay. 1,3-bisphosphoglycerate plus ADP gives 3-phosphoglycerate plus ATP. Phosphoglycerate kinase is reversible. This is your first ATP generating step, and it is substrate level phosphorylation. The phosphate is handed directly from substrate to ADP. No membrane, no proton gradient, no electron transport chain. That is why this reaction works in a cell with no mitochondria and in a tissue with no oxygen. Because it runs twice, it produces 2 ATP per glucose, which exactly recovers the 2 you spent in the first stage. Break even. This is the break-even point. You're now level. Everything after this will be a gain, net gain. 2 clinical hooks. Anaerobic tissues live on this reaction and on step 10 and on nothing else. Red blood cells, the Rappaport-Lou-Behring shunt, bypasses this very step to make 2,3-bisphosphoglycerate. The cell gives us this ATP in exchange for better oxygen unloading. Okay, objective 6. Mutase and enolase. a setup. Pyruvate kinase is the payoff. Grouped because only the last is examinable as a control point. The fluoride grade top details sticks. The last three reactions taken together, steps 8 and 9 are setups. Phosphoglycerate mutase moves the phosphate from carbon 3 to carbon 2. Enolase removes water, producing phosphoenol pyruvate. Pyruvate kinase transfers that phosphate to ADP, giving ATP and pyruvate. Step 10 is irreversible. Why is PEP such a good phosphate donor? Because when it gives up its phosphate, the product tautomerizes from the enol form to the keto form, and that conversion is strongly favorable. PEP has the highest phosphoryl transfer potential of any molecule in metabolism. That usually gets asked on its own. Now, the exam content. Pyruvate kinase is the third control point. It is feed-forward activation by fructose 1,6-bisphosphate, the product of phosphofructokinase 1, telling the bottom of the pathway that material is coming. It is inhibited by ATP and by alanine. In the liver, glucagon-driven phosphorylation inactivates it. Pyruvate kinase deficiency causes chronic hemolytic anemia because the red blood cell has no other way to make ATP. That is objective 7, and it is high yield. And this is one lab detail that shows up in questions as a scenario. Fluoride inhibits enolase. That is why blood drawn form for glucose measurement goes into a gray top tube containing fluoride and oxalate. Stopping glycolysis in the tube stops the red cells from utilizing glucose you're trying to measure. If a stem of the question says glucose, just look out for that particular thing in the stem of a question. If you wrote 4 ATP at the start and you switch to 2 now, you've understood what's going on. So let's look at the tally. Look at what you wrote down. Count out loud. Stage 1, 2. Stage 3, make 4. That's your gross. Your net is 2. If you chose 4, you quoted the gross and skipped your subtraction. So pay attention to that. 0 means you did not consider what happened at stage 3. You forgot the cleavage, and that is twice. Exams usually ask for the net. Check your questions. Check whether it says net. So the answer is B, 2 ATP and 2 NADH. Now we deal with the irreversible steps. 4 bypass enzymes. The mismatch is between PP carboxykinase, which needs pyruvate carboxylase to get there. You're going to be asked about that. 3 irreversible reactions, 3 control points. Learn these 3 and their regulators. Number 1, the hexokinase or glucokinase in liver and beta cells. Glucose to glucose 6-phosphate. It traps glucose in a cell. Hexokinase is inhibited by its product, glucose 6-phosphate. Glucokinase is not. Number 2, the phosphofructokinase 1, fructose 6-phosphate to fructose 1,6-bisphosphate, rate limiting and committed. It's inhibited by ATP and citrate, activated by AMP and fructose 2,6-bisphosphate. This is where insulin and glucagon act. Okay, so let's look at the pyruvate kinase. Phosphoenol pyruvate to pyruvate. It's substrate level phosphorylation with a large negative delta G. Remember, the large negative delta G is about irreversibility. Feed-forward activation by fructose 1,6-bisphosphate, inhibited by ATP and alanine. Always be ready to say the regulator list out loud, because the exam tests the direction and not just the name. Glucose 6-phosphate inhibits hexokinase, but not glucokinase. ATP inhibits both phosphofructokinase 1 and pyruvate kinase. High energy charge shuts down the pathway at both ends. AMP activates phosphofructokinase 1. Citrate inhibits phosphofructokinase 1. Alanine inhibits pyruvate kinase because alanine is one transamination away from pyruvate and therefore signals that pyruvate is already plentiful. And fructose 1,6-bisphosphate activates pyruvate kinase. It's feed forward, the only activator in the list that comes from inside the pathway itself. One structural point when you study gluconeogenesis, because all these three are irreversible, gluconeogenesis can reuse the other 7 enzymes and only needs to work around these 3. Okay, because remember, they're irreversible, so you can't back them up. You have to find bypasses. Because pyruvate carboxylase and PEB carboxykinase are needed, there are 3 irreversible steps, 4 bypass enzymes. Know that. Okay, so check out this question, write down your answer. A saturated enzyme reports nothing because its rate is identical at 5 millimolar and 15 millimolar. Let's compare hexokinase to glucokinase. Same reaction, opposite logic. It has hexokinase first. It has a low Km, approximately 0.1 millimolar. Blood glucose sits around 5 millimolar. What this means is hexokinase is fully saturated at all normal glucose concentrations. It runs flat out all the time. It has a low Vmax, so it cannot be overwhelmed. It is present in every tissue. It is inhibited by its product, glucose 6-phosphate, which is how it stops when the cell has had enough. Its job is to guarantee every cell its own glucose supply, regardless of what the blood is doing. Okay, now let's look at glucokinase. It has a high Km, approximately 10 millimolar, which is right in the middle of the physiological range that blood glucose actually travels through. High Vmax, so it can handle the flood of glucose arriving in the portal vein after a meal. It is found only in the liver and pancreatic beta cells. It is not inhibited by glucose 6-phosphate. It is induced by insulin over hours. Its job is not to feed a cell. It is to report glucose. It's the glucose sensor. Okay, look at hexokinase. It's flat across the physiological range. This is the physiological range of glucose. So hexokinase cannot report glucose. Glucokinase, however, climbs through it, which is what makes it a sensor. A saturated enzyme cannot report anything. because its rate at 5 millimolar is identical to its rate at 15 millimolar. Only an unsaturated enzyme changes its rate when glucose changes. So the sensor has to be the high Km enzyme. Look at the curves. Remember, so there's your Vmax, Km is half Vmax. Look at that. Hexokinase is flat across the physiological range. Glucokinase climbs through it. The curve is the argument. Okay, look at the question. We just talked about this. You guys have this to review so you can see. High Km. The activity must rise as blood glucose rises. That is why the answer is B. Okay. In the beta cell, the rate of glucokinase sets the rate of glycolysis, which sets the ATP concentration, which closes the ATP-sensitive potassium. Now here are the transporters. It's half the center. GLUT2 is high Km and bidirectional, so intracellular glucose in the liver and beta cell equilibrates with blood glucose, which is the only reason glucokinase's high Km can report anything. Low Km GLUT1 and GLUT3 saturate and hold brain and red blood cell uptake constant. GLUT4 is the insulin responsive one in muscle and fat, and GLUT5 is the fructose transporter you meet again. Also, settles the term used back at step 1. GLUT carries neutral glucose, never the charged phosphate. GLUT is the facilitative glucose transporter family, passive, bidirectional carriers, moving glucose down its concentration gradient. No ATP. Each isoform's Km determines what its tissue effectively sees. The ones you are asked about, GLUT1, red blood cells, blood-brain barrier, cornea, low Km, so uptake is constant and guaranteed. GLUT2 found in the liver, pancreatic, beta cells, kidney, and the basolateral side of the intestine. High Km and bidirectional, which is the crucial pairing because it means intracellular glucose in the liver and beta cells equilibrates with blood glucose. Without a high Km transporter, a high Km sensing enzyme would have nothing to sense. GLUT2 and glucokinase work as a unit. GLUT3 is found in neurons, low Km, guaranteed supply of glucose. GLUT4 is found in skeletal muscle and adipose tissue. This is the insulin responsive one. It's stored in vesicles and translocated to the membrane when insulin binds. That is a transporter defect at the heart of insulin resistance. And GLUT5 is the fructose transporter in the intestine and testes, which returns us at objective 8. We'll talk about that later. One tieback. These transporters carry neutral glucose. They do not carry glucose 6-phosphate. That is why phosphorylating glucose at step 1 traps it. Okay, so look at this board question. The distractors are all true statements about some enzyme. A and C describe hexokinase, D describes PFK1, E is invented. Read this stem. 19-year-old woman fasting glucose repeatedly between 1/18th and 1/25th over 2 years, asymptomatic, normal weight, no ketosis, hemoglobin A1C 6.1%. Father and paternal grandmother with the same mild, stable picture, never treated with insulin. Autoantibodies, negative. Sequencing shows that heterozygous loss of function variant in GCK. Which property of the affected enzyme best explains why her hyperglycemia is mild and non-productive? 5 options. 4 of them are true statements about some other enzyme in this lecture. That is exactly how these items are constructed. Commit to one before advancing. Okay. Clinical translation distinguishing the MODY2 from early type 1 or type 2 matters. These patients are often overtreated. Negative autoantibodies plus a strong dominant family history plus mildness is the triad. The answer is B. It has a high Km, so it functions as a glucose sensor, and a partial defect merely raises the beta cell's set point. The reasoning. Because glucokinase reports glucose rather than simply consuming it, losing one functional copy shifts the reporting threshold upwards. Insulin is still released, just a slightly higher glucose concentration. So you get lifelong mild, non-progressive fasting hyperglycemia without ketosis, without progression, and usually without any need for pharmacotherapy. The work that distracts, because that is the transferable skill. Option A, allosterically inhibited by glucose 6-phosphate, is true of hexokinase, not glucokinase. Option C, low Km and saturated at all physiological concentration, is also hexokinase. Option D, the rate-limited committing step in all tissues, is phosphofructokinase 1. That's true, but not for this question. Option E requires fructose 2,6-bisphosphate as an activator, is also phosphofructokinase 1. Every wrong answer is a true statement about some other enzyme in this literature. That is exactly how these items are built, and is why knowing which property belongs to which enzyme matters more than knowing the properties in isolation. Knowing which property belongs to which enzyme. The bifunctional enzyme is the elegant part. One polypeptide, 2 opposing activities. And pK phosphorylation flips which one runs. Fructose 2,6-bisphosphate is the single most testable molecule in carbohydrate metabolism. I'll restate. Fructose 6-bisphosphate, the most tested. Phosphofructokinase 1. This slide is the highest yield regulatory content in carbohydrate metabolism. Okay, so I'll go over it slowly. 5 allosteric signals. ATP inhibits. ATP is also substrate. However, ATP can inhibit. The resolution is that there are 2 binding sites. There's the catalytic site, which has a high affinity, and a separate low affinity allosteric site that only fills when ATP is abundant. Plentiful ATP means the cell does not need to burn glucose, so the pathway is slowed. AMP activates. AMP rises when ATP is being consumed, so it is the reciprocal signal. It relieves the ATP inhibition. This is a dominant control in exercising skeletal muscle. Citrate inhibits. Citrate leaving the mitochondrion signals that the citric acid cycle is saturated and biosynthetic precursors are plentiful. No need to send more carbon in. This is the dominant control in liver. Hydrogen ion inhibits. Falling pH slows glycolysis in muscle, which is a brake preventing lactate from accumulating without limit. And fructose 2,6-bisphosphate activates, the most potent activator of the enzyme and the one that overrides ATP inhibition. Now, fructose 2,6-bisphosphate deserves its own place because it's arguably the single most testable molecule in this subject. It is not a pathway intermediate. It goes nowhere. It is manufactured from fructose 6-phosphate for the sole purpose of being a signal. It is made and destroyed by one protein with two opposing catalytic activities, the bifunctional enzyme. Phosphofructokinase 2/fructose bisphosphatase 2, which activity depends on whether the protein is phosphorylating. Following the logic in the fed state, insulin leads to dephosphorylation of the bifunctional enzyme. That leads to activation of phosphofructokinase 2, which raises fructose 2,6-bisphosphate, which activates phosphofructokinase 1, which drives glycolysis. Insulin, high fructose 2,6-bisphosphate burn glucose. Now during fasting, glucagon raises cyclic AMP, which activates protein kinase A, which phosphorylates the bifunctional enzyme. Phosphorylation switches off the kinase activity and switches on the fructose bisphosphate 2 activity, which destroys fructose 2,6-bisphosphate. Levels fall, phosphofructokinase 1 goes quiet, and gluconeogenesis proceeds. Glucagon, low fructose 2,6-bisphosphate, make glucose. The bifunctional enzyme has tissue-specific isoforms, and in cardiac muscle, phosphorylation has the opposite effect. It activates the kinase activity, raising fructose 2,6-bisphosphate and accelerating glycolysis. Adrenergic stimulation of the heart should burn more glucose, not less. The liver isoform is the one you default to unless the stem says heart. One thing worth remembering, because I explain why this molecule exists at all, fructose 2,6-bisphosphate simultaneously activates phosphofructokinase 1 and inhibits fructose 1,6-bisphosphate, the opposing gluconeogenic enzyme. One molecule, two reciprocal effects, and therefore guaranteed that glycolysis and gluconeogenesis never run at the same time in the same cell. Running both simultaneously would be a futile cycle that burns ATP and accomplishes nothing. Lactate production is not failure. It is the solution to an NAD+ problem. The lactate itself is recycled, not discarded. Glycolysis has produced pyruvate and NADH. The NADH is a problem before it is an asset, because unless NADH+ is regenerated, step 6 stops and the whole pathway halts. Everything on this slide is a solution, describes a solution to that problem. Aerobic route first. Pyruvate enters the mitochondrion. Pyruvate dehydrogenase converts it to acetyl-CoA, and that enters the TCA cycle for complete oxidation to carbon dioxide. The cytosolic NADH cannot cross the inner mitochondrial membrane. It needs a shuttle. And there are two, and which one a tissue uses changes the ATP yield. You have the malate aspartate shuttle, which operates in the heart and liver. It delivers the electrons as NADH in the matrix, which enters at complex 1. That's worth roughly 2.5 ATP per NADH. The glycerol 3-phosphate shuttle operates in skeletal muscle and brain. It delivers the electron as a FADH2, which enters this complex 2, bypassing complex 1. It's worth roughly 1.5 ATP. It's faster but lower yield. When you see a total ATP yield of glucose quoted as 32 in one source and 30 in another, that difference is a shuttle. Do not memorize a single number. Understand which shuttle produced it. Now let's look at the anaerobic route. With no oxygen or with no mitochondria, pyruvate itself becomes an electron sink. Lactate dehydrogenase reduces pyruvate to lactate using NADH, regenerating NAD+, which allows glycolysis to continue. Notice what the cell gets and does not get. It keeps the 2-substrate level ATP. It gets no oxidative phosphorylation and no value from the NADH because the NADH was spent buying back NAD+. 2 ATP per glucose is the entire anaerobic yield. Which tissues do this obligately? The mature red blood cell, because it has no mitochondria, the lens and the cornea, which are avascular. Exercise in skeletal muscle shock states and hypoxia use it transiently. This reaction is fully reversible, and the direction depends entirely on the NADH to NAD plus ratio and on the concentration of pyruvate, which is why a cell that becomes reoxygenated can run it backwards and recover the pyruvate. And in tissues where oxygen is plentiful, the same enzyme converts lactate arriving from elsewhere back into pyruvate for oxidation. And do not call lactate a waste product. It is exported to the liver, converted back to pyruvate and then to glucose, and returned to the muscle. That is the Cori cycle. Clinical translation lactic acidosis is what you see whenever pyruvate cannot proceed forward. Shock and hypofusion, pyruvate dehydrogenase deficiency, thiamine deficiency, and metformin all produce it by leaving pyruvate nowhere to go but lactate. 2,3-BPG compensation in pyruvate kinase deficiency is a favorite exam point. These patients tolerate anemia better than the hemoglobin number suggests. Contrast with hexokinase deficiency, where 2,3-BPG falls and patients feel worse. The red blood cell is a tissue that makes glycolysis clinically visible. So this slide earns disproportionate exam weight. The mature red cell has no mitochondria, none. Therefore, glycolysis is its only source of ATP, and lactate is the obligate end product. That ATP has a specific job. It runs the sodium-potassium ATPase and maintains membrane deformability. Lose the ATP and the cell stiffens, loses its ability to squeeze through the splenic cords, and is destroyed by splenic macrophages. That is why a defect in a glycolytic enzyme presents as a hemolytic anemia, and specifically an extravascular one. The pentose phosphate pathway also runs in the red blood cell, and it is essential because it supplies NADPH for glutathione reduction. It makes no ATP. The red blood cell has exactly one energy pathway. When the hemolysis looks like in the laboratory is worth stating, because STEM describes it rather than name it. Unconjugated hyperbilirubinemia and jaundice, elevated lactate dehydrogenase, low haptoglobulin, the marrow compensates splenomegaly from the organ doing the destroying. Because the destruction is extravascular, you do not see hemoglobinuria. Now the shunt. The Rappaport-Leuer-Behring shunt diverts 1,3-bis-phosphoglycerate, the product of step 6, away from phosphoglycerate kinase, converting it instead to 2,3-bisphosphoglycerate. The cell gives up 1 ATP for every molecule diverted. That's the real cost, and the red blood cell pays it deliberately. What does it buy? 2,3-bisphosphoglycerate, which binds in the central cavity of deoxyhemoglobin and stabilizes the toned state, the tense low affinity state. The oxygen dissociation curve shifts to the right and hemoglobin unloads more oxygen in the tissues. 2,3-bisphosphoglycerate rises in chronic hypoxia, in anemia, and at high altitudes. It is an adaptive trade of ATP for oxygen delivery. If in pyruvate kinase deficiency, the block is at the bottom of the pathway. Everything upstream backs up, including 1,3-bisphosphoglycerate. More substrate for the shunt means more 2,3-bisphosphoglycerate, a right-shifted curve, and better oxygen unloading. So these patients tolerate their anemia better than the hemoglobin number predicts. If you contrast that with hexokinase deficiency, where the block is at the top of the pathway, nothing accumulates downstream. 2,3-bisphosphoglycerate falls, the curve shifts left, and those patients feel worse at the same hemoglobin. Same anemia, opposite curve shift, entirely because of where the block sits. Sometimes students remember 2,3-bis BPG binds hemoglobin, but reverse which state it stabilizes. The stem. Okay, this board question. 4-year-old boy, lifelong jaundice, splenomegaly, hemoglobin 8.4 with reticulocytosis. Smear shows no spherocyte and osmotic fragility is normal. That pairing is there to exclude hereditary spherocytosis. Enzyme assay shows markedly reduced erythrocyte pyruvate kinase activity. Symptoms are milder than the hemoglobin predicts, and 2,3-bisphosphoglycerate is elevated. So you have 5 options. Option D is a trap. It states the mechanism backwards, and it catches people who remember that this molecule binds hemoglobin, but not which state it stabilizes. So pay attention to that. What state it stabilizes. In hexokinase deficiency, the block is at the top of the pathway. So 2,3-BPG falls, and those patients are symptomatically worse at the same hemoglobin. Same anemia, opposite curve shift. The answer is B. Glycolytic intermediates proximal to the block accumulates, raising 2,3-bisphosphoglycerate, which right shifts the oxygen curve and improves tissue unloading. Okay. Let's look at the distractors. D is a trap. We talked about that. It says that 2,3-bisphosphoglycerate stabilizes the R state. Wrong. That's backwards. 2,3-bisphosphoglycerate stabilizes the T form, the deoxy state, and decreases affinity. Fix the direction now. 2,3-bisphosphoglycerate, T state, right shift, more unloading. Option A says the PPP generates 2,3-bisphosphoglycerate. It does not. It makes NADPH and ribose 5-phosphate. Option C invents an inhibition of lactate dehydrogenase that does not exist. And option E says the red blood cell compensates by upregulating oxidative phosphorylation, which anyone who knows the mature red blood cell has no mitochondria, should eliminate that answer in a second. The lactate to pyruvate ratio is a discriminating lab value. Blocks at PDH raise it. Blocks within glycolysis proper do not. Thiamine before glucose is a genuine bedside rule. Glucose loading is a thiamine deficient patient precipitates Wernicke. So there are 4 clinical entities. Learn each as a triad: the lesion, the presentation, the discriminating lab. Pyruvate kinase deficiency, autosomal recessive, most common enzyme defect of glycolysis. Red blood cell ATP depletion, rigid cells, extravascular hemolysis, splenomegaly, jaundice, often presenting in the newborn period. Upstream intermediates accumulate, so 2,3-bis phosphoglycerate is high and the oxygen curve is right shifted. No spherocyte, normal osmotic fragility. Pyruvate dehydrogenase deficiency, most commonly X-linked, affecting the E1 alpha subunit. Pyruvate cannot become acetyl-CoA, so it is diverted to lactate and to alanine. Remember alanine, because pyruvate and alanine are 1 transamination apart. The presentation is congenital lactic acidosis with neurological damage from infancy. So treatment is ketogenic diet. Give the patient ketogenic substrates that produce acetyl-CoA downstream of the block. The ketogenic amino acids you will come to, but remember them. They are lysine and leucine. Arsenic. Distinguish it from arsenate. Arsenic, arsenite, inhibits lipoic acid and therefore blocks both pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, since both of them use lipoate. The result is lactic acidosis. Clinical, we use garlic breath, rice water stools, vomiting, peripheral neuropathy. I won't be asking you for clinical answers. You must use the clinical information to understand the biochemistry. Thiamine. Okay. Cofactor failure. Wernicke encephalopathy. We'll skip this. Students, you guys met galactose as a C4 epimer. Here it becomes UDP galactose 4 epimerase and a neonatal emergency. Essential fructosuria versus HFI is a classic benign versus lethal pairing. 2 topics on the side. Hormonal control first. What matters here is a 2 time scale. Insulin acts on 2 clocks. Over minutes, it raises fructose 2,6-bisphosphate through the bifunctional enzyme, which is allosteric and instantly reversible. Over hours, it induces transcription of 3 enzymes: glucokinase, phosphofructokinase 1, and pyruvate kinase. These are the same 3 irreversible enzymes from objective 2. The control points are the control points at every level. Glucagon does the reverse on both clocks. Protein kinase A activates fructose bisphosphatase 2, lowering fructose 2,6-bisphosphate within minutes, and it also directly phosphorylates and inactivates hepatic pyruvate kinase. Over hours, the induction effects reverse. The liver commits fully either to consuming glucose or to exporting it, never both. Okay, the other dietary hexosis. And here's where you have inborn errors. Fructose. Dietary fructose is absorbed on GLUT5 in the liver. Fructokinase converts it to fructose 1-phosphate, and aldolase B cleaves that to trioses, which join glycolysis at the level of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Below phosphofructokinase 1, that is where that occurs. Fructose bypasses the committed rate-limiting regulated step entirely. There's no ATP or citrate feedback on fructose entry. So the carbon floods in unregulated, and much of it ends up as acetyl-CoA and then fat. That is why fructose is lipogenic. 2 fructose disorders. Deficiency of fructokinase causes essential fructosuria. Fructose appears in the urine as a reducing sugar. The patient is asymptomatic and no treatment is needed. Deficiency of aldolase B causes HFI, which is dangerous. Fructose 1-phosphate accumulates and traps the cell's free phosphate, which shuts down glycogenolysis and gluconeogenesis, and the result is severe hypoglycemia, vomiting, jaundice, liver damage after ingesting fruit, juice, honey, or sucrose. Onset is when the infant is weaned onto those foods. Treatment is dietary elimination of fructose, sucrose, and sorbitol. The mild one is missing the kinase. The severe one is missing aldolase B. Galactose comes from dietary lactose, which lactase splits at the intestinal brush border into galactose and glucose. Galactokinase phosphorylates galactose to galactose 1-phosphate. Galactose 1-phosphate uridyl transferase GALT then converts it towards glucose 1-phosphate, and UDP galactose 4 epimerase interconverts the 2 sugars at carbon 4, which is the only carbon at which galactose and glucose differ. Galactokinase deficiency is relatively mild. Galactose accumulates and is reduced by aldose reductase to galactitol, which causes infantile cataracts. The infant fails to track objects and develops cataracts without systemic illness. GALT deficiency is classic galactosemia, and it is a neonatal emergency. Failure to thrive, vomiting, jaundice, and hepatomegaly. Cataracts, intellectual disability, characteristic predisposition to E. coli sepsis in a newborn. Treatment is exclusion of galactose and lactose from the diet, which in practice means removing breast milk and standard formula. A lab point that links these 2 disorders: galactose, fructose, and lactose are all reducing sugars. So all 4 conditions can give a positive urine reducing substance test, while the glucose-specific dipstick, which uses glucose oxidase, stays negative. A positive reducing substance with a negative glucose dipstick in an infant should direct you to these pathways. Okay, the left figure explains why fructose is lipogenic. Okay, because the right figure is the classic galactosemia, the GALT step. The fructose figure shows the entry point. Trace it. Confirm for yourself that fructose enters below phosphofructokinase 1. That geometry is the entire explanation for why fructose escapes regulation. The galactose figure shows the 3 enzymes in order: galactokinase, GALT, and the epimerase. Locate GALT on it. That step is classic galactosemia. Two things to learn. First, the 2 times scales. F26BP moves within minutes and is fully reversible. Enzyme induction takes hours and is what makes a fed liver structurally different from a fasted one. Students conflate them constantly. In the footer, F26BP activates PFK1 and inhibits FBPase1, so one molecule guarantees the two pathways never run at once. That is the futile cycle argument. FBPase1 we'll study later. Note the three enzymes named on both slides are the same three. Glucokinase, PFK1, pyruvate kinase, the control points. And the hormone figure consolidates the 2 times scales. Allosteric control through fructose 2,6-bisphosphate within minutes. Transcriptional induction over hours, acting on the same three enzymes. This slide is the spine of the whole week. Every remaining carbohydrate lecture is one of these 4 hours. Okay, so therefore, there you have glucose 6-phosphate sits at a 4-way junction, and glycolysis is only one of the 4. Okay, glycolysis. Onward through glycolysis to pyruvate for ATP, 2 in glycogen via glucose 1-phosphate and UDP glucose. 3 into the pentose phosphate pathway for NADPH and ribose 5-phosphate. Remember that. PPP, NADPH, and ribose 5-phosphate. No ATP from that route. And the 4th, dephosphorylation by glucose 6-phosphate phosphatase back to free glucose, which is then exported to the blood. Okay, glucose 6-phosphatase exists only in liver and kidney. Muscle does not have it. That is why muscle glycogen can supply the muscle itself, but can never raise your blood sugar. The muscle physically cannot release free glucose. And the disease attached to that enzyme is von Gurk disease, glycogen storage disease type 1. Without glucose 6-phosphatase, the liver can neither complete gluconeogenesis nor release glucose from glycogen, so the patient presents with severe fasting hypoglycemia. Hepatomegaly, lactic acidosis, hyperuricemia, and hyperlipidemia. Every one of those findings follows from glucose 6-phosphate being unable to cell. That single fact answers a surprising number of questions. Okay, 5 items. If you retain only 5, these are the 5. The yield. Net 2 ATP, 2 NADH, 2 pyruvates per glucose. Gross is 4 ATP. Know both numbers and know that the exam asks for net. 2. The 3 irreversible steps. Hexokinase or glucokinase, phosphofructokinase 1, and pyruvate kinase. These are the control points in the reactions gluconeogenesis must bypass. 3. Fructose 2,6-bisphosphate, the most potent activator of phosphofructokinase 1. Insulin raises it, glucagon lowers it, and the same molecule inhibits fructose 1,6-bisphosphatase, so the 2 pathways can never run at once. 4. The red blood cell. No mitochondria, so glycolysis or nothing. Pyruvate kinase deficiency gives hemolytic anemia with high 2,3-bisphosphoglycerate and a right-shifted oxygen curve. 5. Hexose entry. Fructose enters below phosphofructokinase 1, escaping the committed step control, which is why it is lipogenic. Hereditary fructose intolerance is dangerous. Essential fructosuria is benign. Okay. Four diseases, each one a lesion at a control point. Pyruvate kinase deficiency. Okay. The red cell has no backup. Losing the last ATP producing step costs it its membrane. Chronic hemolytic anemia with 2,3-bisphosphoglycerate piling up behind the block. And with DY2, glucokinase is a sensor, not merely a kinase. So loss of function allele resets the beta cell's threshold and gives mild, stable, fasting hyperglycemia. Lactic acidosis, shock. Pyruvate dehydrogenase deficiency, thiamine deficiency, and metformin all leave pyruvate with nowhere to go but formation of lactate. Hereditary fructose intolerance. Fructose skips the checkpoint and allulose B deficiency traps phosphate as fructose 1-phosphate. Hypoglycemia and vomiting after fruit or sucrose. Okay, so you have glycolysis, you make pyruvate, now pyruvate moves on. It has arrived at the mitochondria. And the pyruvate dehydrogenase enzyme is the gateway from pyruvate to acetyl-CoA. And that is the end of this lecture. Thank you very much and good luck. Remember your enzymes, where they're found, what are the cofactors, what are the yields of energy products like ATP and NADH, which reactions are reversible and which are not.